Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and I'm your biology educator. Good evening. Yes, I am audible and visible. And guys, as you can see, so we have restarted the All India mock test series, guys, and it will not be these tests won't be weekly now. They'll be twice in a week. That is on Sunday and every Wednesday 2 to 5 p.m. Is it clear? Make sure that now you don't miss this opportunity. You know what? At times in life we thought that okay, what if we get another chance? And now we have got this great chance. So please make sure that now we don't we utilize it to the best. And yes guys, you are going to take these All India mock tests every Wednesday and Sunday. Good evening everyone. Is that clear? So guys, with these so you have to enroll. The link is given in the description box. Also they are pinned in the chat, I think. Okay, it will be pinned in the chat very soon, and you will get the real NEET exam experience. Also you will get a rank prediction that out of 22 lakh students who are, or more than 22 lakh students who are appearing in the tests, so what is your rank going to be? So make sure that you take these tests regularly and don't miss even a single test. And then on top of the test, analysis is very, very important. So video solutions will be available right after every test. And guys, yes, this is Re-NEET Bounce Back series class 12. We are going to complete Botany part 3 today. So guys, in this session, we are going to complete 4 chapters: morphology, anatomy, cell structure and cell cycle. So give me a thumbs up that you're all set and we can start with the session. So give me a thumbs up that you're all set and we can start. Really? Veenu bhai? Stop this. The voice is too loud already. Alright guys, now I'm going to quickly start with the session. Once again, I'm reminding you, you have to enroll for this. Every Wednesday and Sunday you have to take this test. The test is starting from 24th of May. That is coming Sunday. Alright, now starting with the first chapter, which is first chapter of the day, which is anatomy of angiosperms. Are we all set? I am all ready with my flowcharts. Quickly let me know guys. Is that clear? All right. Now, let's begin with the chapter of anatomy of flowering plants. And now guys, in this chapter, some part is not given in new NCERT, but we have to discuss it. So make sure that you you subscribe to the Telegram channel and you download the notes. Is that clear? So now see, first of all, we are going to talk about the meristem. We are going to talk about the 2 types of tissues, 2 types of plant tissues, which are number 1, meristematic tissue and permanent tissue. Meristematic tissue and permanent tissue. Is that clear? Now we already know that meristematic tissue is the one which can divide. It is actively dividing tissue. So the cells in this tissue are actively dividing cells. While the permanent cells, they don't divide. They have lost the ability to divide. Then what do? So guys, these cells, this tissue is important for every function in the body. So these cells don't divide, rather they specialize for a specific function. They have specialized for a specific function. Are we clear? So there are these 2 types of plant tissues. Now we must know that both these tissues, they are interconvertible. Actually, it is the meristematic tissue which gets converted into permanent tissue, and we've already discussed that. What do we call this process as? I have discussed this aspect in plant growth and hormones. Yes, So this aspect is known as differentiation. This process is known as differentiation. Is that clear? And then now on the basis of, see, I can divide meristem into, meristem can also be divided into, okay, different types. So types of meristem. So types of meristem. Now see, meristem can be of two types based on number one, origin. On the basis of origin. On the basis of origin. And second, on the basis of location. So there can be different bases on according to which we can divide this meristem. Now first of all, see, on the basis of origin, we have two types of meristem, which is primary meristem and secondary meristem. Right? We have already discussed about this in the chapter of plant growth and hormones, how primary and secondary develops. Is that clear? Now let me talk more about the types of meristematic tissue on the basis of location. So guys, on the basis of location, this is of three different types, which is one, apical meristem, then we have intercalary meristem, and the third is intercalary, and the third one is lateral meristem. So apical, as the name says, apical means it is present at the tips, So we have two tips, shoot tip and root tip, and eventually we have two types, shoot apical meristem and root apical meristem. Is that clear? Root apical meristem and shoot apical meristem. Now root apical meristem is basically covered by the root cap. So root apical meristem is not exactly terminal. So this becomes subterminal. While subterminal because of root cap. While shoot apical meristem is present on the tip, it is terminal. Are we clear? So both these, basically the apical meristem is going to help us in increase of length and depth. So it helps in the increase of the length of the shoot and depth of the root. Please tell me these points are clear. Are we clear here? Is that clear? Right? Are we clear here? I don't really understand Sigma, and there are other students. Why do you keep on spamming? Like you have got one aspect just to keep telling me that voice is low. Where is my voice? Why? How is my voice low? Please change your appliances. This is so irritating. In the middle of the class, you are just distracting the teacher by saying again. Now, just because I'm checking your chat so that nothing misses out, you're just spamming uselessly. Stop this nonsense totally. Just concentrate. If you can't hear, then why are you here? You will spend the entire three hours here just wasting your and my time. And I am not going to look at your chat at all. Useless. Next we have, guys, intercalary. Now where is intercalary present? It is present at the nodes. It is present at the nodes. So what is the function? What is the function of intercalary meristem? This intercalary meristem gives rise to the mature parts. It gives rise to the mature parts. All the mature parts like leaf, flower, bud, tendril, thorn, right? All these structures which are arising at the node, they are arising because of intercalary meristem. Are we clear? Right. And then we have lateral meristem. Lateral meristem is present at the sides. And what is the function of lateral meristem? It helps in increase of width, or you can say girth of the stem. and this is present in the form of cylinders. Hashtag present in the form of cylinders. Is that clear? So these are the three types of meristem. Now coming on to yes, permanent tissue. Let's talk about permanent tissue. So permanent tissue is the one which has lost the ability to divide, but now it can only perform specific functions, and that's what we need really, right? Apart from growth, we want some specific functions to happen. So now see, what is a tissue actually, guys? So when we say tissue, tissue is a group of cells, right? With common origin, basic structure same, and performing the same function, right? But when we come on to permanent tissue, we get to see a little anomaly here. Now see, we have simple tissue, and we have complex tissues. Now simple tissues are the one which consist of similar cells, while this consists of different cells, and still we call them tissue because of the common origin, and overall function is same. Is that clear? The mother cells, the basic origin of the cells is same. Now see, so simple tissues are further of three types: parenchyma, collenchyma, and sclerenchyma. The main function of parenchyma is, guys, storage. Apart from other functions, collenchyma is going to give us flexibility and strength, and sclerenchyma is responsible for rigidity because of this lignin. And because of lignin, these sclerenchyma cells are going to become dead, while these are living and living. While these are earlier living, but as the lignin deposition increases, they will become dead, right? And then in complex tissue, guys, we have two types. We have xylem and phloem. Xylem is for the transport of water and minerals, while phloem is for food conduction. So we can call complex tissue as the vascular tissue also, because the two types of tissues present in this category are both meant for conduction, and they are the vascular tissue. Please tell me these points are clear, right? Now moving on to the details. See here, parenchyma, the main function is storage. It is the most abundant tissue in the plants. Like in animals, it is always the areolar connective tissue, while in plants it is the parenchyma. The shape is isodiametric. They can be spherical, round, oval, polygonal, or elongated. They have thin cell walls, and which is made up of cellulose. Now the cells are always very closely packed with few intercellular spaces. Is this point clear? Now see the functions. The most important function is storage, but other than storage, it is also meant for secretion. It will absorb as well. It will photosynthesize, provide buoyancy, and sometimes it is going to provide support also. Is that clear? Now there are two modifications of parenchyma depending upon what they are storing. Now see, this is parenchyma. Okay. So this is parenchyma, guys. So what is parenchyma? It is that type of parenchymatous tissue which stores air. It stores air. So what is air going to do, guys? Air is going to provide buoyancy. So this is found in the aquatic plants. Which can float, so that they can float for floating purpose, like water lily, that is lotus. Then, guys, we have Sclerenchyma. Right? So what is Sclerenchyma going to do? Sclerenchyma is going to contain chlorophyll. So the main function of Sclerenchyma would be photosynthesis. What will be the function? Photosynthesis. So basically, the palisade mesophyll layer of leaves that represents the Sclerenchyma. Are we clear till here? See, palisade mesophyll layer represents the Sclerenchyma. Are these points clear to all? So guys, this is about parenchyma that you must know. Now, moving on to collenchyma. This is a second type of simple permanent tissue. Cells can be of different size, shapes, oval, spherical, or polygonal. They are present below the epidermis and usually form the continuous layer, or they can be found in patches. right? Now thickened deposition, other than cellulose guys, they have pectin and hemicellulose. Other than cellulose, they have pectin and hemicellulose, and these depositions are found not all around the wall. Let's see, let's say if this is a cell, if this is the cell, on the radial walls, on the radial wall we have some depositions. These are the depositions of pectin, cellulose and hemicellulose. Are we clear? So what happens, these depositions will provide strength also, and when the deposition is absent, it will contribute towards flexibility as well. Are we clear? So this is the tissue which provides flexibility and mechanical support. So the two locations where we need both of these qualities, one is midrib of the leaf and second is the petiole. So you want the leaf to be able to fan, to provide that fanning movement for the cooling, as well as it should have strength so that while fanning it doesn't break. Is that clear? So midrib and petiole are possessed with, they contain Sclerenchyma. Please tell me these points are clear. Are we clear? Yes. Moving on now to the third type of simple permanent, which is Sclerenchyma. Now the cells can be of two different shapes. Do you see? These can be long, tapering from the ends, right? These types are known as fibers. These types are known as fibers. This is one modification. And second modification of these Sclerenchyma are stone cells, or we also call them sclerids. Now, as the name suggests, so these are circular in size. These are rounded in size, and the whole space is filled with lignin. Now see, Sclerenchyma, they can be long, narrow or oval. They are thick and they are lignified. Lignified means there is lignin deposition, guys. Now wherever there is lignin deposition, lignin is going to end the protoplasm. There will be loss of protoplasm. The whole space of the cell will be all taken over by lignin. So we say there is loss of or obliteration of lumen. Do you see? This space could have been filled by cytoplasm, but now it has all been taken up by this red colored lignin. Now there are some pits present for the transport. That's it. Is that clear? Now if the cells are these rounded or oval, we call them stone cells, while these tapering cells will be known as fibers. So remember, wherever you find this word fiber, xylem fiber, phloem fiber, they will be sclerenchymatous in nature. Are we clear? Now lignin is going to make it hard. It is going to give a lot of rigidity and provide mechanical strength. Now where are these cells present? So already I told you, fibers, they will be found in xylem and phloem. They will be found in xylem as well as phloem, right? But the stone cells, see, the stone cells are found in the fruits of the nuts or the fruit walls of nuts, pulp of fruits like guava, sapota and pear. You know, there are certain fruits which have that gritty feeling while you eat. So all that grittiness is because of stone cells. Is that clear? If there is wood around you, then you know how that, you should know that that is how Sclerenchyma feels like. It is so hard. You know, the brown husk of coconut, that's what Sclerenchyma feels like. Is that clear? So that is all Sclerenchyma. Third, it is found in the seed coat of legumes as well as in the tea leaves. So when the leaves are dried, so that dryness, that dry leaves, they contain Sclerenchyma. Please tell me these points are clear. Is that clear? Now moving on to the complex permanent tissue. First, we are going to start with xylem. So guys, xylem is commonly known by the name of wood. It is majorly a dead tissue. Why? Because see, xylem contains 4 different components. It has xylem vessels, xylem tracheids, xylem fibers and xylem parenchyma. Now out of these 4 components, it is only the xylem parenchyma which is living, while all other are dead. Fiber, Sclerenchyma, dead. Vessels, Sclerenchyma, dead. Tracheids, Sclerenchyma, dead. Is that clear? So out of 4, 3 are dead. So that is why it is mainly a dead tissue. We know the function of xylem. Now the movement of water in xylem is always in upward direction, unidirectional movement. What is the function? Conduction, as well as it is going to give a lot of mechanical support. Is it clear? Now let us look at the 4 components, starting with vessels, guys. Now vessels are a characteristic feature of angiosperms only. That's how angiosperms derive their name. Angion means vessel. So out of these 4 components, these 3, tracheids, parenchyma, fibers, they will be present in gymnosperms. See, I have already revised plant kingdom with you and we have discussed it there also. But these structures, vessels, they happen to evolve only during the angiosperms. Is that clear? They are seed bearing and the fruit is enclosed. Sorry, seed is enclosed within the fruit. We know what angiosperms mean, but the word angio comes from vessels. Is that clear? And then tracheids, dead, Sclerenchyma. Now these are pointed and with tapering ends. Now see, there are 2 diagrams given in NCERT. How do you differentiate? So vessel is more like a cylindrical structure. It is not tapering. It is not pointed from the end. The one with the pointed end is called tracheid. So how do we remember? T for tracheid, T for tapering. Tapering means pointed. So T for tracheid, T for tapering. The one with the pointed end is a tracheid. That's how you identify. And this was a PYQ, guys. This was a PYQ already. Yes? Are we clear? Are we clear? Yes, Sanskar, the part which is not visible, I'm already writing down. So there is some issue with the PDF, probably it has got cut. So whatever is missing, I'm writing down personally. Okay? All right. Now, this side, see, xylem fibers. Fibers, I've told you, always made up of sclerenchyma, and because they are sclerenchymatous, so the lumen is obliterated, means the lumen gets vanished. They can be septate or aseptate. Parenchyma is living. Now, what is the function of xylem parenchyma? It stores food, water, and then tannins. Remember that. Now, just after this, we are going to talk about phloem. Now see, xylem parenchyma was storing food, water and tannins. Now look here, what phloem parenchyma, this is the phloem parenchyma. What does it store? This stores food, resins, latex and mucilage. Is it clear? Now how do we remember this? So in order to remember, there is a trick which is permal. It is permal. So phloem parenchyma will store resins, mucilage and latex. While in case of xylem parenchyma, it is tannins and food and water. Will you be able to remember? Now guys, this phloem parenchyma, this one is absent in monocots. It is absent in monocots. Please remember. Right? Now coming on to fibers. Fibers are dead. Fibers are always dead. See, while xylem was majorly dead, phloem is majorly a living tissue. Why? Because except for fibers, rest 3 components are going to be living. They are all living. So 3 out of 4 are living, hence it is living tissue. Is that clear? Now see, fibers are sclerenchymatous. Now phloem fibers are known by the name of bast fibers. Now remember, there is a commercial importance of phloem fibers or bast fibers. The fibers of bast fibers of jute, hemp and flax are meant for commercial use. So these baskets and carpets and the ropes, they are made from the bast fibers of these plants. Is that clear? Now moving on, and these fibers are always unbranched, long and pointed, be it phloem fiber or be it a xylem fiber. Now coming on to sieve tubes. Now see, again, while discussing gymnosperms, I told you sieve tubes and companion cells, these are only found in angiosperms. These are angiosperm characters. While in gymnosperms, guys, instead of sieve tubes, we have sieve cells. We have sieve cells in gymnosperms. Is this point clear? And instead of companion cells, gymnosperms will have albuminous cells. Is this clear? Already discussed, just revised. Now let's talk about sieve tube. What does sieve mean? Sieve means the one which is used for filtration. So see, this is the sieve plate. So the end of the sieve tube, this is a sieve tube, the end has sieve plates. This is a sieve plate. Now, sieve tubes don't have their own nucleus, hence they are enucleated. So in order to control and regulate the sieve tubes, companion cells are always present in the vicinity of sieve tube. This is the companion cell. You will always see companion cell giving company to the sieve tube for control and regulation, because sieve tube doesn't have its own nucleus. Are we clear? Right? Are we clear? All right. Moving on now, guys, to the next component, which is okay. Yes, which is tissue system. Let's talk more about tissue system here. This is fine. Now see, so we have talked about the tissues, meristematic tissue as well as the permanent tissue. Now let me discuss the tissue system. So overall, any part of the plant can be divided into three major tissue systems. So let's say this is a root or a stem. So the outer part is known as epidermis, the outer layer. Now epidermis might have some modifications like root hair or stem hair. Then or similarly in case of leaf, there will be epidermis, upper epidermis, lower epidermis, right? It will have cuticle, it will have stomata. So all these structures present outside, on the outer side, consisting of epidermis, their modifications, their secretions, right? They are all coming under epidermal tissue system. Now see, there are three tissue systems. One is epidermal tissue system, made up of parenchyma, always single layered, and major function is protection. Outer, so it will provide protection. What are the components? It consists of epidermis, the appendages, some depositions, and the stomatal apparatus. Is that clear? Now, appendages could be root hairs or the stem hairs, right? So, stem hairs are also known as trichomes. What is the deposition I'm talking about? I'm talking about cuticle, and I'm talking about the stomatal apparatus. And when we say stomatal apparatus, guys, it contains guard cells. Then the opening known as stomata. And third, the subsidiary cells. These three components together form the stomatal apparatus. Please tell me these points are clear. Please tell me these points are clear, right? So there are four components which together form the epidermal tissue system. And note down what type of cells are majorly present here. We'll discuss the details also, but first understand the components. Then talking about vascular tissue system. So vascular tissue system already, as the name suggests, consists of vessels. That means like conducting structures, xylem and phloem. So it consists of xylem and phloem. So the function is, of course, conduction. Now, other than epidermal and vascular tissue, rest everything that I have colored in purple, that is all ground tissue system. So you will see the major part of any organ is made up of ground tissue. Is that clear? So you've heard of the layers called cortex, endodermis, pericycle, pith, medullary rays. They are all basically forming the ground tissue. And now these can be formed of parenchyma also, collenchyma, as well as sclerenchyma. What are the functions? The functions can be a variety, like transport, providing strength, giving then also storage of certain things. Is that clear? And they form the major bulk, means a major part of any organ is formed of this. Are we clear till here? Please tell me. Are we clear, guys? Yes, yes, yes. Now, moving on to the details here. Moving on to the details. Let's see. Epidermal tissue system. I told you outermost protective, but since it is outer layer, it will absorb also, it will secrete also, and it will prevent transpiration as well. Now, wait a second. Something has happened. Okay, never mind, we'll adjust. So, guys, epidermal tissue system, these are the functions discussed already. Now, it consists of single layered epidermis, right? Epidermis. What is it made up of? I told you epidermis is always made up of parenchyma, and it is single layered. It is single layered. But at times it can be multi-layered. That is in case of desert plants, in case of stem and leaf, to prevent transpiration. So that will be a xerophytic adaptation. Otherwise, normally epidermis is always single layered. Now, talking about the epidermal appendages. What are the appendages? In case of roots, guys, in case of roots, you might find root hairs. In case of stems, you can find trichomes. Stem hairs are also known as trichomes. Is it clear? These are the epidermal appendages because they develop from, because they develop from this outer layer called epidermis. So, now see, in case of root, guys, in case of root, this cell can show an outgrowth, and this outgrowth will be known as a root hair. This is how a root hair looks like. Root hair is always unicellular and it is always unbranched. While the stem hair, these are trichomes. See, the trichomes, the stem hair can be multicellular as well as unicellular. They can be unicellular as well as multicellular. They can be branched or unbranched. Branched or unbranched. Are we clear? Please tell me. Are we clear about the differences between this root hair? The function of root hair is, guys, to absorb. The function is absorption. While the function of stem hair is always either to reduce transpiration or they are secretory. They might secrete something or they can be secretory. They don't absorb anything like root hairs do. Are we clear? Please tell me, are we clear here? Yes or no, guys? All right, moving on now to the next, which is the vascular tissue system. First of all, we'll talk about the vascular tissue system. Now see, the vascular tissue system is made up of two components, that is xylem and phloem. That is made up of xylem and phloem. Let me discuss it here. The vascular tissue system. So the vascular tissue system will consist of vascular bundles, right? These are found in the vascular tissue system. Now these vascular tissue system or vascular bundles can be divided into three different categories on the basis of three different bases. Now see, on the basis of arrangement of xylem and phloem, we have two types of vascular bundles. Is that clear? We'll talk about it. Then on the basis of presence of cambium, again we have two types of vascular bundle. Is that clear? And then we have the third type, that is on the basis of position of protoxylem. Position of protoxylem. Are we clear? Please tell me. So there are different bases. These are on the basis of number 1, arrangement, number 2, position of protoxylem, and number 3, presence of cambium. Yes? Now see, guys, on the basis of arrangement of phloem, so there are two possibilities. One, that xylem and phloem are present alternate to each other like this. Right? So when xylem and phloem are present alternate to each other, presence alternate xylem and phloem, right? So in this case, we call such arrangement as, or such vascular bundle as, radial vascular bundle. So we say the xylem and phloem are alternate to each other or present on different radius, present on different radius. Is that clear? Now this condition is always seen in roots. R. We don't have to remember triple R, we have to remember RR. Radial in arrangement in roots. Is that clear? Then second is when xylem and phloem are present in the same bundle. Xylem and phloem on the same bundle. So in that case, so we have phloem on the outer side, xylem on the inner side, like this. But when they are present on the same radius, so on same radius, or you say in the same bundle, in the same bundle, such vascular bundle, guys, is known as conjoint. And this is seen in stem. Is that clear? So on the basis of arrangement, we have two types of vascular bundles. Is that clear? Now, on the basis of position of protoxylem. See, protoxylem is a type of primary xylem itself. So the parts or the elements which are formed in the initial stage are called protoxylem, and which are formed later are known as metaxylem. So these elements of protoxylem, they are small in size as compared to metaxylem, which are larger in size. Now I'll show you how. Let's say this is a vascular bundle and this is another vascular bundle. I'll compare and tell you how each one looks like. Now, if the protoxylem, the smaller particles are on the inner side and the larger particles on the outer side, like this. So this small particle, this represents the protoxylem. Is that clear? While there can be another situation where the protoxylem is on the outer side, like this. Like this. So when the protoxylem is on the outer side, protoxylem outer, so this condition is known as X-arch. You know, X is someone who's out of life. So remember X. Protoxylem on the outer side, right? And then protoxylem on the inner side, this is called end-arch. So you just have to see the location of protoxylem and nothing else. And how do you observe that in the given diagram? Smaller size. The smaller element is protoxylem, the larger element is that of metaxylem. Are we clear? Please tell me. Are we clear here? Hi Sagna. Yes, of course you can join the session. You're most welcome. You can always go with the flow and revise the leftover later on. Now guys, see, this exarch condition is always found in the root, while endarch situation will be seen in stem and leaf. Is this clear? Now, talking about the presence of cambium. Now you have to remember this trick, dock. So guys, in dicot, there is open vascular bundle because the cambium is present. Cambium is present. Can you remember it? Yes or no? Can you remember this dock? D-O-C. This is the way to remember. Now, cambium we know helps in the secondary growth, right? So there can be two situations. Let's draw the vascular bundle here. So this is one vascular bundle. This is another vascular bundle. And this is the xylem, this is the phloem on the outer side. Again, xylem and phloem. Now see, in between the two, xylem and phloem, if there is a layer of cambium present, if cambium is present, such vascular bundle is called open vascular bundle. It means cambium is present and it is open to secondary growth. So secondary growth will take place here. Secondary growth is also positive here, right? While in this case, guys, there is no cambium. So cambium is absent. No cambium means no secondary growth. So such vascular bundles are called closed vascular bundles. Yes or no? These are the two types. Are we clear? Now, the point to be noted is open vascular bundles are found in dicots, be it dicot root. Well, the arrangement of cambium is different in case of root, but it is open in case of root. This is the vascular bundle of stem, dicot stem. While closed is found in monocot, be it root or stem. Is that point clear? So monocots will not show secondary growth because of the lack of cambium. Are we clear, guys? So these are the different types of vascular bundles, right? So this was little not clear, so I've drawn this. Now guys, coming on to the structure of root. Let's draw the structure of root here. Now see, let's say this is the structure of transverse section of root, okay, and dicot root. So the outer layer, this is the outer layer. What is the outer layer known as? This outer layer is known as epidermis. Now epidermis of root, guys, is also known as epiblemma. And we know that epidermis is always made up of parenchyma. It is always single layered, right? Now, these cells can show an outgrowth known as root hair. This forms the root hair. Is that clear? Then, similarly, these cells will be present and at times they will give an outgrowth known as root hair. Are we clear? So, see, the function of root hair, wait. Now, see, these two structures, root hair and epidermis, together, guys, they are going to form the epidermal tissue system of root. Is that clear? This is always single layered, monolayer and made up of parenchyma, right? Root hairs are always unicellular and they're formed as an outgrowth of these parenchyma cells, right? Now, since the origin of root hair is from the outer cell, it is just formed from the outgrowth of this outer cell, we say that root hairs are exogenous in origin. They are exogenous in origin. Is that clear? Exogenous in origin, right? Now, there will never be a presence of cuticle. If cuticle is found in roots, cuticle is totally absent. If cuticle is present, guys, root won't be able to absorb water because it is a waxy coating. Imagine you put a layer of oil around the root, it will not be able to absorb water. Yes, so cuticle is absent in case of roots, be it dicot or monocot. First, let's see the general structure, then I'll bring in the specifications of dicot and monocot root. Are we clear? Now, on the inner side, guys, on the innermost side, like this, here, let's say here, yes, here we have vascular bundles. So here we have vascular bundles, like this is xylem and this is phloem, xylem and phloem. We have just read root has a radial type of arrangement, yes, and this is xylem and this is phloem, and the xylem phloem are in alternate format, xylem phloem, yes. Now this xylem and phloem, they are differentiated by the presence of these cells, and there was a question this year in the match of following. Remember, there was this question, what are these cells known as which differentiate xylem and phloem? These are known as, this tissue is known as conjunctive tissue. This tissue is known as conjunctive tissue, again parenchyma, but it separates xylem and phloem. Is that clear? This is all conjunctive tissue, right? Now, in the center here, there is present these parenchymatous cells, and these parenchymatous cells are known as pith. This is known as pith. Now, pith is always made up of parenchyma and it is always going to store food. Is that clear? So, the pith in case of dicot root, okay, let's talk about dicot and monocot later. First, just understand the structures. Is that clear? So, in the center we will have pith, right? Now, just outside this vascular bundles, we have one or two layered thick layer known as pericycle. So, we have this layer known as pericycle. Are we clear? This is pericycle, right? This is pericycle. Now, very important, guys, very important. Pericycle is again made up of parenchyma, but it has thick-walled parenchyma. It is thick-walled parenchyma. What is the function? It is this pericycle which will give rise to the lateral roots. It will give rise to the lateral roots, and it is going to give rise to cambium when needed. So these are the two functions, lateral roots and cambium. Now see, lateral roots means, see, we have one taproot and then we have secondary and tertiary roots. So that secondary and tertiary roots originate from pericycle. So they are not an outgrowth like root hairs are. They're coming from inside of the root, that is from the pericycle. Hence, the lateral roots are endogenous. Root hairs are exogenous, lateral roots are endogenous. Do we understand this point? Please tell me. Yes, guys, do we understand this point? Yeah? All right. Now, outer to this, we have this layer known as endodermis, made up of these barrel shaped cells. This layer is called endodermis. This is endodermis. Now guys, endodermis here is again made up of parenchyma, but this parenchyma is going to be thin-walled. This is thin-walled parenchyma. Now what is so special about this endodermis? This endodermis is lined by this waxy deposition which is called suberin. So there is suberin deposition seen in here. Suberin is deposited and suberin makes it water impermeable. And these suberin depositions are known as Casparian strips. These are known as Casparian strips. Do we see here? Yeah. So, and on the outer side, guys, here, filling the entire space, compactly arranged, this is all cortex. This is all cortex. And what is cortex made up of? Again, thin parenchyma. Now remember, and with very few intercellular spaces, few intercellular spaces. Is that clear? Very few intercellular spaces. Are we clear? Now remember, endodermis is always the innermost layer of cortex. It is called the innermost layer of cortex. Innermost layer of cortex. Is that clear? Now, how does the water transport occur? First, let's see this. So water will be absorbed through these root hairs and it will enter into the cortex, and from cortex, guys, from cortex it has to go into the deeper layers. But then we have this endodermis. Endodermis has water impermeable layer, that is called Casparian strips. So water cannot move into the layers with deeper layers through endodermis. Why? Because of suberin. Suberin is not, it is water impermeable. So water doesn't get through the endodermis like that. In between the endodermis layer, guys, there are some special cells. Let's say these, which are not deposited by suberin, and we call these cells as passage cells. And these passage cells, as the name says, form a passage for the water from cortex into the deeper layers. Are we clear? Are we clear here? Right? So this is a general structure, general transverse section of a root. Now, what are the differences between a dicot root and a monocot root? The difference comes at two levels. One is at the vascular bundle. Now see, in case of, let's say if this is a dicot root, in a dicot root, the vascular bundles, as I've drawn here, how many? One xylem phloem, 2, 3 and 4. There are 4, 2 to 4 in number. Not more than 4. Is that clear? Second, see, be it any root, two things are confirmed, that the condition will be radial and exarch. But because it is a dicot root and we have to remember DOC, remember? So the vascular bundle will be an open type. It is an open vascular bundles because cambium will develop later from the pericycle and it will help in secondary growth. Are we clear? Please tell me. So there are 2 to 4 vascular bundles, open type and radial and exarch. Now, if there are two vascular bundles, we'll call it diarch condition, triarch condition, tetrarch condition, but not beyond that. Is it clear? Second difference comes here, pith. This is a food storing tissue, food storage, and since it is involved in storage, so it is made up of parenchyma again. Now, pith in case of dicot, guys, in case of dicot, hashtag, this is small and inconspicuous. NCERT writes this word, inconspicuous means underdeveloped. But if you look at the diagram, it looks the opposite. While comparing monocot and dicot root, it will look the opposite, but the theory confirms it, that pith is small and inconspicuous. Please tell me these points are clear. Is that clear? But instead of dicot root, had this been a monocot root, then what would be the difference? What would be the difference here? So guys, in monocot root, instead of 2 to 4 vascular bundles, the number of vascular bundles would have been more than 6. So this condition is known as polyarch. This condition is known as polyarch. This is open because cambium will develop. Here it is closed vascular bundle, no cambium. No cambium means no secondary growth. Are we clear? Right? Then another difference comes around pith. Here it is small and underdeveloped, for which NCERT says inconspicuous, while the monocot pith is large and well developed. Please tell me these points are clear. Yes, guys. Please tell me all these points are clear and are we clear? Yes. So NCERT has clearly written, yeah, that these Casparian strips are present on the radial and tangential walls. Right? Cool. Now, there is another term which is called stele. Something is happening today. Okay, never mind. Alright, guys. So, another term is stele. So what is stele, guys? Stele is a term used for all the tissues lying inner to endodermis. So say if this is endodermis, what is lying within the endodermis is pericycle, vascular bundles and pith. These three components together known as stele. Clear? Now coming on to the transverse section of a dicot stem. Now see very carefully. Yeah. So the outermost layer is always known as epidermis. So this is just a cross section. The outermost layer is called the epidermis. Clear? What is epidermis, guys, made up of? Epidermis is always made up of parenchyma. Right? And how many layers of parenchyma? Just single layer. Now since it is a stem, it may have cuticle layer. It may have stomatal apparatus also. Till it is a young green stem, it will have stomatal apparatus, right? And then it also has these hair which are known as trichomes. Is that clear? Do we see epidermal hair? These are nothing but trichomes. And we have read that the trichomes are multicellular. Do we see it? They have not shown the branch type of trichomes, but they can be, right? Now, inner to epidermis, we have cortex. Now see, guys, the cortex in case of dicot stem is well developed. It is well developed. Together, these three layers, hypodermis, this, they form the cortical layer. This forms the cortex. So cortex in case of dicot root is well differentiated and well developed. So there are three subzones. Is that clear? These are the three subzones, which are number 1, hypodermis. Second, we call it cortical layers. And the third, as the name suggests, it is endodermis. Is that clear? Now, cortex in case of root was made up of parenchyma, but cortex here in the dicot stem is made, this hypodermis, the cortex is differentiated into 3 layers. The outer one is known as hypodermis, middle is cortical layer, and then we have endodermis, right? So you see these layers here. So this whole structure, this whole pink region, this represents the hypodermis. And what is hypodermis made up of? Collenchyma. It is made up of collenchyma. Then cortical layers are made up of parenchyma. So this is the cortical layer, second layer. And this whole green thing represents the cortical layer. Is that clear? And then guys, we have the innermost layer of cortex, which is endodermis. Endodermis now is known by the name of, it is also known as starch sheath, because here the parenchyma cells are filled with starch grains. Since they are rich in starch grains, so we call this layer as starch sheath. Is this clear? Inner to endodermis, we have pericycle. Now remember, now look at this pericycle, guys. The pericycle is present. This is pericycle. So pericycle is not continuous. It is in the form of semi-lunar patch. And just below pericycle, we have phloem. So we say, or NCERT says, it is present above phloem. In root, guys, it was made up of parenchyma. Here, it is made up of sclerenchyma. It is made up of sclerenchyma. This is made up of collenchyma. This is made up of parenchyma. This is also parenchyma with starch grains. This is made up of sclerenchyma. Right? Is that clear? Then just below this, we have phloem, and then inner to it, we have xylem. Now, do you see the arrangement of vascular bundles here, guys? Now, in case of dicot root, it is a typical feature that the vascular bundles are in the ring arrangement. They show ring arrangement, and this is a characteristic of dicot stem, not found anywhere else. Please tell me, are these points clear? So the vascular bundle shows they are in the ring arrangement. We see a ring being formed, and center to that, in the central region, there are parenchymatous cells which constitute pith, and so pith is very large in case of dicot stem. And what does pith do? It always stores food. So what is it made up of? Always parenchyma. Are we clear till here? Yes or no? And now guys, in between, see, in between any two vascular bundles, these cells, this whole space, there are present medullary rays. Now what are medullary rays? This is nothing but parenchyma. We had a question this year. It is made up of parenchyma that differentiates, it separates xylem, it separates the two vascular bundles. Yes or no? These are radially arranged. So the parenchyma is arranged like this, radially arranged parenchyma, which forms or which separates the two vascular bundles. Please tell me these points are clear. Now, another thing about the vascular bundles. See here. Vascular bundles here are arranged in the form of a ring. Now, since it is a stem, guys, it will have end-arc condition. It will have conjoint condition. See, xylem and phloem present in the same bundle. First phloem, and then inner to the phloem we have xylem. So they're present in the same bundle, hence it is conjoint. It shows end-arc condition, and while deciding whether it is end-arc or exarch, what do we notice? We notice the position of only protoxylem. So see, protoxylem, the smaller elements on the inner side, hence these are inner, hence it shows the end-arc condition. Protoxylem on the inner side, hence it is end-arc condition. And since it is a dicot, guys, so do you see a layer of cambium right here? So the presence of cambium makes it an open vascular bundle. It is an open vascular bundle. Please tell me these points are clear to all. Please tell me all these points are clear to you. Quick, quick, quick. Are all these points clear to everyone? Let's have a quick thumbs up break. Yes, guys. Quickly let me know. Are we clear? Yes? Are we clear here? Yes, ma'am? Perfect. All right. Let's move on now. Moving on to the next topic. That is monocot root. Now see, unlike dicot root, look at the vascular bundles here. The vascular bundles here are all scattered. The vascular bundles here, these are all vascular bundles. These are all vascular bundles. So vascular bundles are all scattered. They're not in the ring arrangement. Is that clear? Now, starting from the outer direction, so we have outermost epidermis. So I told you, since it is a stem, it can have cuticle, it can have stomatal. It is a stem, yes. So the younger ones can have cuticle and stomatal apparatus. Look at the layer of cuticle. Clear? Then, in this monocot stem, guys, the cortex is not well developed. Cortex is reduced. Like in dicot stem, it was well developed. In monocot, this is a monocot stem. In monocot stem, it is highly reduced. Right? Now see, the cortex is only represented by hypodermis. And here, hypodermis, do you see? The hypodermis forms a very thin layer and it is made up of sclerenchyma. Rest of the ground tissue is not differentiated into different layers like endodermis or pericycle or pith. No, it is not differentiated. So we simply call it ground tissue. Hashtag the ground tissue not differentiated. Like in root and dicot stem, we have seen different layers known by different names, performing different functions, made up of different tissues. But here it is not differentiated. It just consists of parenchymatous cells, and within this ground tissue, the vascular bundles are scattered in GT, that is ground tissue. Are we clear? Now these are all the vascular bundles. Do you see the smaller ones are on the periphery? Smaller on the periphery, while larger ones in the center. Is that clear? Now this is just an enlarged version of this vascular bundle. Do you see these structures? Yes, these structures which look like eyes. These are not eyes, guys. These are the water cavities. These represent the metaxylem, yes, and it represents the water cavities. Yeah, sorry, not this. This one, protoxylem lacuna. These are known as water cavities. So basically the parenchyma got destroyed and resulted in water cavities. Are we clear? Please tell me these points are clear. And since, guys, this is a monocot and we have read about the process called C4 cycle, we have seen the anatomy also. Most of the monocots have C4 cycle. They are mostly C4 plants, not all of them. So all the C4 plants will have Kranz anatomy. Now see, do you see there are these thick bundle sheath cells, and bundle sheath cells are always sclerenchymatous. Now can you relate why bundle sheath cells were carbon dioxide chambers? Because their wall is so thick it cannot let the CO2 escape through it, and that is why it becomes CO2 chamber, and that is why Rubisco shows carboxylation in bundle sheath cells. Do we see that? So these are the bundle sheath cells present around the vascular bundle. Please tell me these points are clear. Anyone with any doubt? Please tell me if there is any doubt. Right? Can we move on now to the next? So as talking about the dicot leaf, see, dicot leaf, the outer layer is known as epidermis. Moving here. So we are going to talk about the dicot leaf here. So dicot leaf is known as dorsiventral leaf. Dorsiventral leaf, because the dorsal and ventral side are well differentiated. So we know the dorsal side is very shiny and green, while the ventral is not, right? So see, here we will see that there is upper epidermis. Upper is known as adexal, and then there is lower epidermis known as abaxial. And on the lower epidermis we have more stomata and no cuticle, while on the upper epidermis there is less stomata and more cuticle. which makes it shiny, right? This is adexal epidermis. So there are two layers, adexal and abaxial, and now can you differentiate between the two? Yes? Wait a second, guys. So I don't know whether you are listening or not. My Alexa is listening already. All right, guys. Moving on now. Now see, there is the, see, this is the vascular bundle, the xylem and phloem. This is the vascular bundle, and there is a layer of sclerenchymatous cells present in, known as bundle sheath cells. So this shows the Krenz anatomy. In dicot, we are looking at the Krenz anatomy. This might have been a C4 plant. Yes. Now listen. Okay. Now in between the vascular tissue and the epidermal tissue, the entire rest of the layers, these layers are known, this whole layer is known as the ground tissue. This represents the ground tissue, and it is differentiated into upper mesophyll layer and lower mesophyll layer. Lower is made up of spongy mesophyll and the upper is the palisade mesophyll. Basically, they are both parenchyma cells. And which type of parenchyma does this palisade represent? It represents Sclerenchyma because it contains lots of chloroplasts. Each cell has around 20 to 50 chloroplasts, and this is where maximum photosynthesis is going to take place. Yes, and in spongy parenchyma, guys, we do see the air cavities, which is going to help in diffusion of gases. Yes or no? And also transpiration. So this is how a dicot leaf looks like. But in case of monocot leaf, now see, in case of monocot leaf, you don't see any differentiation between upper and lower epidermis. So adexal as well abaxial, they look alike. Both have cuticle, both have similar number of stomata. There is no distinction. So that is why we call monocot leaf as isobilateral leaf. We call it isobilateral leaf. Please tell me these points are clear. Yes? Then we have this xylem, we have this phloem, this is the phloem, right? And then we have, so the entire mesophyll is of same type, that is spongy type. It consists of spongy mesophyll only. It is not differentiated into palisade and that. It is only made up of spongy type. Do you see? All cells are similar. So you can't differentiate, hence it is isobilateral. Is that clear? Then there are these substomatal cavities and the stoma or the stomata on both sides. So this is the structure of monocot leaf. Please tell me these points are clear. So guys, I've already discussed the Krenz anatomy with you. So bundle sheath cells surrounding the vascular bundle. Clear? Now, there are very important cells present in monocots, and very important topic. This is already a PYQ. Please listen carefully. These cells are known as bulliform cells, also known as motor cells. Motor means something that moves. So these cells don't move, but they cause the movement of the leaf. They cause inward curling of the leaf. See, let's say this is an isobilateral leaf, and around the midrib, guys, there are these special cells present. These cells are called motor cells. So what happens? Basically, see, these cells are specialized along the central vein of monocot leaf. They remain turgid when there is enough water, and when they are turgid, when they are all filled with water, the whole lamina, the leaf is all exposed, the lamina is exposed. But when there is water stress, it loses water, they become flaccid, they wrinkle up, and the lamina curls inward. So the leaf is going to fold inwards. Curling of leaf, not on the outer side, but on the inner side, like that. So it will help in reducing the surface area, and this is one method of checking the transpiration. Please tell me these points are clear. Are these points clear? Right, guys. So this is a flow chart. Please use this flow chart. Take a screenshot, guys. This flow chart will help you in revising the entire topic. Quickly take a screenshot. Is that clear? Is that clear to all? So this is for your revision. I have revised it in detail already. Now guys, moving on to the next chapter. This next chapter is that of morphology. Next chapter is morphology. Now, let's see. Morphology is a study of external structure of anything, but we are talking about morphology of angiosperms, flowering plants. So if we talk about a typical flowering plant, the plant body is divided into two parts, which is root system and shoot system. The root system is underground, develops from the radicle. What are the main functions? It is anchorage and absorption, right? While the shoot system is the aerial part, that means exposed in air, develops from plumule, and functions can be conduction, photosynthesis and reproduction. Are we clear? This is basic. Now talking about them one by one. Okay, let's discuss about roots. Now there are there are different types of roots, guys. So first of all, we have tap root. We have a tap root. Then we are going to talk about adventitious roots, and third, fibrous roots as well. Now let's understand what each means. So tap root means it develops from the radicle. The primary root will give rise to the lateral root. So this is the tap root, and from here will arise these secondary, tertiary roots, and which will eventually give rise to the root hair. So there is one primary, then secondary, then tertiary, and so on. So this type of root system is called tap root system, and it is a typical case of dicots. For example, gram, mustard, all these are tap roots. All they have tap roots because they are dicots. Now moving on to the fibrous roots. Look here first. Fibrous roots, fibrous roots are present in monocots. For example, sugarcane, wheat, rice, maize, bamboo, they are all fibrous. They all have fibrous roots because they are all monocots. Now in case of fibrous roots, see how do they look like? They look like, let's say, like this. They look like this. This is how fibrous root looks like. Now the point to be noticed that in case of fibrous roots also, the beginning of root system started with the radicle. Okay, but the one which came from radicle, that was primary root, and that primary root was very short-lived. Primary root was short-lived. Primary root comes from radicle only, but that was very short-lived, and it was replaced by adventitious and fibrous roots. They were replaced by these fibrous roots. And these fibrous roots, they arise from the base of the stem. From the base of stem. Please tell me these points are clear. Is that clear? They rise from the base of the stem. Now, in addition to tap and fibrous root, guys, we have this third category called adventitious roots. Now, adventitious roots, the main function of adventitious roots is they are supportive. They provide extra support. Okay, so they don't arise from radicle, they arise from part other than radicle. See, arise from parts other than radicle. Clear? Now, they are further of two types. One is stilled and second is banyan root. So, banyan roots are found in case of hanging roots are found in case of banyan, while stilled roots are found in case of sugarcane, bamboo, monstera. Let me tell you how do they look like. Now see, let's say this is sugarcane, right? These are the nodes. Now we do know that sugarcane grows very tall. So in order to provide extra, see, so right. So in order, and since it is a monocot, it will have these fibrous roots first of all. Now in order to provide extra support, do you see this last node? Do you see this last node? So there are some roots arising from the last node and then going into the soil like this. Do you see? From the lower node, these roots will arise and they will go deeper into the soil to provide extra support. These are called stilled roots. Where do they arise from? From lower nodes. Arise from lower nodes. Right? While in case of banyan tree, so these are the lateral branches, right? The extra roots, they will arise from these lateral branches, and then they grow into the soil. So this is how the hanging roots, they are hanging from the top branches. These are called hanging roots, and these are found in banyan. Are we clear? Please tell me, are we clear, guys? So these are the two types of adventitious roots, right? Now, moving on to the next, that is regions of root. Now, regions of roots have already been discussed in the chapter of plant growth and development. Let's have a quick revision, guys. We know that there are three parts of the regions of the root. One is the meristematic region. Then we have region of elongation or zone of elongation, and we call it maturation zone or zone of root hair, right? So what are the functions? See, so in maturation, differentiation, presence of root hairs, and then elongation, cells will enlarge because of increased vacuolation. And what is the zone of meristem doing? Active cell division, cells are small, thin, and contribute towards the primary growth. And then we have a root cap secreted by these cells of meristem. Yes, so we have already discussed this. Now root cap, we know it is thimble-like, secreted by meristem, protects the meristem. So root meristem becomes subterminal. We have already discussed it. Now coming on to the root modifications. Now look very carefully, guys. See very, very carefully. Yes, please. Please let me know that these points are clear. Yes, guys, quickly. Please let me know these points are clear. Now, talking about the root modifications, see, root modifications are for different purposes. Some roots, they will store food, like carrot, radish, turnip, sweet potato, and asparagus. Now, it can either be taproot or adventitious, both can store food. So, these three examples are the tap roots which are storing food, while these two, sweet potato and asparagus, are the examples of adventitious roots which are storing food. Are we clear? Now, there are others which are meant for support. So, just now we have talked about stilt roots and hanging roots. So, stilt roots and hanging roots, I'll write down here. So, hanging roots are meant for extra support. Stilt roots meant for extra support. Where are they present? They arise. See, hanging roots arise from the upper branches. Stilt roots arise from the lowest node. We have already discussed this, and these are the examples here. Now, the third type of modification of roots is, guys, for respiration. So, we call them respiratory roots. We call them respiratory roots, or by the name of pneumatophores. Now, these roots are only found in those plants which are growing in very salty, saline or marshy areas. For example, rhizophora. So, we know that root is supposed to be going towards the gravity. So, root is typically, see, a typical root, let's write down here. See, a typical root is always positively geotropic. That means it goes towards the gravity. It is positively hydrotropic, goes towards the water, but it goes against the light, so negative phototropic. But in case of respiratory roots, guys, we see that these roots, instead of growing deeper, they start growing out of the water, out of the ground, because they are getting suffocated in the marshy and the saline conditions. So they come out of the ground and grow aerially upwards. They grow, yeah, so they are aerial in nature. They are negatively geotropic. So these have special openings for the respiration. We call these openings as pneumatothords. Are we clear? Are we clear, guys? These are respiratory roots known as pneumatophores. Please tell me these points are clear to everyone. Are we clear? Yes? Also guys, please understand that although Re-NEET is going to occur, definitely going to happen, and we are doing all this for Re-NEET exam, but at the same time, I don't want the students who are preparing for NEET 2027 to feel left out. No, your syllabus has already started, your batches have already started, so make sure that you quickly enroll in those batches and start with your preparation. My batches have already started in class 12. Is that clear? So quickly enroll. There is a link given in the, it is pinned in the chat also. Enroll and get your classes started. Don't wait for Re-NEET. It is for 2026. So you don't have to delay your preparation for 2027. And by the way, please see certain videos. I have uploaded some, the update of NTA. NTA is planning, NTA and the government, they are planning lot of reforms. So those who are looking forward to NEET 2027 have to prepare very strategically. So it's going to be different. What exactly will come out of these discussions happening in the parliament, we don't know yet, but something is cooking up. So get ready for NEET 2027 in the best possible way. So don't waste your time. Just start your preparation now, okay? And do that very seriously. Not like this chatting in the class and spamming and this is useless. So in order to avoid all this chit chat and any kind of distraction, we have those batches. Let's join and sit together and study for 2027. All right, guys, moving on now to the next, that is stem. So stem, I told you, stem grows away from the gravity. So it shows negative geotropism. It grows towards light. So it shows positive phototropism, right? It is the ascending part, develops from the plumule. It is green when young, brown and woody when it's old. Then what are the functions? What are the functions? The functions are support. Number one function is support. Second function is that it conducts water and food. Then storage. Then support can be given through tendrils. Then also it provides protection. So protection is given in the form of thorns. So there are so many functions which a stem is performing. Also sometimes they help in asexual reproduction, that is by the means of vegetative propagation. So there are so many structures. See, stem is represented by these structures. Stem has nodes, it has internodes. There are axillary buds present in the nodes, and then the buds which are present at the tip, they are known as terminal buds. So these are the structures present on the stem. Are we clear? Now talking about the stem modification. Now, like in case of root, stem can also store food. Is that clear? Stem can also store food. So it stores food like here. Wait. For storage, stem can also store food. For example, potato. Right? Potato stores food. So that is why we eat. So that is also stem modification. Then it provides support also in the form of tendrils, like in case of gourds. All the gourds have stem tendrils. Remember that. Then gourds are cucumber, pumpkin, watermelon. Clear? Then we have, so stem tendrils are present in which all plants? That example, these are very, very important things. So see, you have to learn it clearly that which plants have stem tendrils, while which ones have leaf tendrils. So write them together and learn them as a point of difference, guys. So remember, leaf tendril, in Hindi we call leaf as patta. So P for patta, P for P, right? So leaf tendrils are present in pea, beans. While stem tendrils, as I just told you, they're present in gourds. Second, it is found in watermelon. Third, it is found in grapevines. That is grapes. Grapevines also have stem tendrils. Is that clear? So cucumber, pumpkin and yeah, cucumber, pumpkins, these are gourds. All the gourds, bitter gourd, bottle gourd. So pumpkins. All of them, all these guards have stem tendrils. Please tell me these points are clear. So the points where you can get most confused, guys, always write them together. Always study them together. Always study them together. Are we clear? Are we clear here? Yes? Have you written them separately on the sticky notes if you are doing it in your NCERT, or if you're writing in your copy, then write them separately at the top of the page. So that's how you always tackle the tricky things. Now coming on to the third important part of the plant, which is leaf. Yes, guys. Now see, leaf is a lateral part, because if this is a stem, so it is going to arise from the sides. So it is a lateral part. It is dorso-ventrally flattened. It is a flattened structure. Where are leaves present? They are always present at the nodes. They always originate from the apical meristem. So do you see at the tip of the stem, there are some baby leaves present? So the meristem keeps on growing upward and the leaves are left behind. So they give rise to the broad structure. So this is a leaf primordia, right? So these are the baby leaves. So new leaves are always at tip. Such arrangement is known as acropetal arrangement. Is that clear? Now every leaf will have three parts. One is the leaf base, second is the petiole, and the third is lamina. While petiole may or may not be present, but leaf base and lamina are definitely present. Lamina is the leaf blade. Is that clear? Now see, this is the node from where the leaf is arising. This is the point of attachment, which is known as leaf base, right? This stalk of the leaf is known as petiole, and this broad area, this exposed surface, is called lamina or the leaf blade. Now remember, there is always, now this area is called axil, right? There is always a bud at the base of the leaf, always a bud, which is called axillary bud. Are these points clear? Always a bud at the axil of leaf, hence it is called axillary bud. Now let's talk about the base first. Now the leaf base can be of three different types. First, it can be stipule. Now see, this is the point of attachment. So this is the leaf base. So from the base, guys, at times these two very thin, they are quite hairy actually, very thin leaf-like structures arise. These structures are known as stipules. Not present in all, of course. They can be seen in some monocots. So these are stipules. Is that clear? Not mono, they can be found anywhere actually, not necessarily in a particular type. So these are stipules. And while we are studying the families, we will see this example. Let me know in the comment in which family do you see stipules. Now, second is the pulvinous. So when the base is swollen, the swollen base is known as pulvinous. Is that clear? Now, pulvinous is seen in case of citrus. Now, talking about the leaf sheath. So what happens when the base is going to surround? So let's say this is a leaf and this is the base. So the base of the leaf is going to surround the stem, right? So when the base surrounds the stem like this, so this is known as leaf sheath, and it is a characteristic of monocots. It is a characteristic of monocots. Please tell me these points are clear. Now, again, talking about the leaf modifications. Talking about the leaf modifications. All right. First of all, the leaf modification which is meant for support, just discussed. So those curly structures meant for climbing called tendrils. So if the tendril arises from the axil area, it is a stem tendril. If it arises from, let's say, if the tip of the leaf gets converted, right, or it comes from here, that is from the petiole. So these are leaf tendrils. Where are they present? P and B. They are meant for protection also. So protection, as well as they are not really protection, they are meant for reducing the water loss, reducing transpiration. They are meant for reducing transpiration. So that's when the leaves get reduced. So reduced form of leaf is known as spine. right? And they reduce transpiration. So this is a characteristic of xerophytes. Do you see cactus? So we all have seen cactus. So do you see these flat structures? These flattened structures are called, these are the stems. These are the stems. The leaves are reduced in the form of spines. So we use the term spine when it is a modification of leaf, and we use the term thorn when it is a modification of stem. Is that clear? So these are the reduced leaves. So here in this case, the stem will become photosynthetic. Is that clear? Now talking about insectivorous plants. Now insectivorous plants, we know they have their leaves, they perform photosynthesis, but they are partially heterotrophic. They make up for their nitrogen needs by ingesting these insects and small animals, right? So in order to trap those organisms, they have these some special leaf modifications. So pitcher plant will have that pitcher. So all those structures are made up of, or they are modifications of leaves. Then we have scale leaves. So we all have eaten onion and garlic. So onion, the part that we eat is basically leaves, but these are the fleshy leaves. These are the fleshy leaves. They store food, and that is why we eat. But on the outer side, guys, do you see that dry cover that we peel off? That area is a scaly leaf. And then we have phyllodes found in acacia, in which the petiole is expanded to perform photosynthesis. So let's say this is the stem, and here actually the petiole will have expanded like this, and the real leaf is very small. This is the expanded petiole, which has become photosynthetic. This is seen in case of Australian acacia. Now guys, next point about leaves is to talk about the arrangement of veins in their lamina. So if we talk about the arrangement of veins and veinlets, we have two types of arrangements. One is reticulate, that is network-like. This is a typical case of dicots, but not dicots only. Or if I say all dicots have reticulate venation, this will be a wrong statement. All is wrong. There are exceptions in which monocots also show reticulate, but we don't have to talk about it. Then parallel venation. Now see what happens in reticulate actually. First let's discuss reticulate more. So in reticulate we see the center one. This is the main vein, and this main vein is known as midrib, while these ones are the veins and the veinlets. Do you see there is this thick network formed? So these are the veinlets. So this network arrangement is known as reticulate venation. While in parallel, guys, there might be one central, but all the others, they run parallel to these. It is difficult to differentiate which one is the primary and the secondary and so on. So this is parallel. While in case of banana also, this is also a parallel type of venation. So there can be one prominent midrib, but rest all of them, they go parallel to each other. This is also a parallel venation. Now this is a typical case of monocots, and yes, banana is a monocot, but not all monocots. Exceptions are always there. Please let me know that these points are clear. Right? Now moving on to the types of leaves. We have types of leaves, guys. We have types of leaves, right? So we have two main types. One is simple and second is compound, guys. So simple leaf means lamina is not divided. It is entire. Not divided means it is entire like this. And in case there are some incisions means there are some cuts, then the cuts don't reach the midrib. So midrib is far and incisions are quite away from them. Incisions do not reach the midrib. And then remember, there will always be, as I said, an axillary bud at the base of the leaf. Is that clear? Now talking about the compound leaves. See, compound leaves are a further two types, pinnately and palmately. See, first of all, how do you identify whether that green structure is a leaf or a leaflet? Please understand, how do you identify? You always look out for the axillary bud. If the axillary bud is present here and not here, not here, so it means these are not leaves, these are leaflets. And the structure ahead of this axillary leaf, sorry, axillary bud. So anything that is ahead of axillary bud is an entire leaf. So this complete is a leaf, but a compound leaf, and the small structures are leaflets. So such arrangement is called pinnately compound leaf. And now see, if this would have been an entire leaf, then this center would be the midrib. So we call this midrib here as rachis. So in case of pinnately compound, the midrib is known as rachis, and around the midrib, the lamina has broken down into smaller leaflets, and the bud is present at the base of the leaf, not at the base of leaflets. Please look at it carefully. Now, see, examples are neem and rose. In case of palmately compound, see what happens. So here we have a bud, and this is the stalk of the leaf, right? And this is the tip of the petiole. The stalk of the leaf is called petiole. So this is the stalk, tip of the petiole, and all the leaflets have arranged themselves at the tip of petiole like a palm of the hand, So at the tip of the this is like palm of the hand. That is why we call it palmately compound. What do we call it? We call it palmately compound. Please tell me these points are clear. And the example is silk cotton. The examples are very, very important in this chapter. Now talking about phyllotaxy. Let's talk about phyllotaxy. Yes, guys. Yeah. All right. Now talking about phyllotaxy. What is phyllotaxy? It is the arrangement of leaves on the stem. It is the arrangement of leaves on the stem. So guys, we have three types of phyllotaxy. One is alternate, second is opposite, and the third one is whorled. Now see the arrangement. In alternate, at every node, we just have one leaf. One node, one leaf. This is alternate. In opposite, do we see? At every node, there are two leaves. 1, 2, 1, 2, 1, 2. So from every node, two leaves are arising. And then whorled. So at one node, there are so many leaves. One node, many leaves. This is called a whorled arrangement. Look at the examples. So alternate, China rose, mustard, sunflower. Opposite, we have calotropis and guava. And for whorled, we have alstonia. Please tell me these points are clear to everyone. Yes, guys, quickly let me know. Are these points clear? Yes, are these points clear to all? All right, moving on now to the next part, which is called inflorescence. Guys. Now, what is inflorescence? Inflorescence, guys, is the arrangement of flowers. It is the arrangement of flowers on the floral axis. Right? Now, see, we have two types of inflorescence, basically. One is called racemose and the second one is called cymose. Okay. Now what is racemose? In racemose, guys, the tip continues to grow. See here. I'll draw it here. So let's say if this tip continues to grow, right? This tip will continue to grow and the lateral branches arise here. So basically, this one is the older flower. So the new branches are arising at the top. So the new flowers are at the tip, new at the tip or near the tip. So this is called racemose. Tip continues to grow and the flowers, new flowers at the tip. So this is in the acropetal succession. While in cymose, guys, what happens? The tip will stop growing and the flowers will be arranged in the basipetal. That means the younger ones will be present or they arise from the base. The younger ones will arise from the base. Like how? So let's say if this was the stem, this tip will stop growing and terminate into a flower. So now when the tip will stop growing, Now the lateral branch will grow ahead. And again, this will stop growing and modify into a flower. Right? Then again, the base, another growth occurs from the base of this, and it will again stop growing and grow into a flower, terminate into a flower. Now see, these ones are the new, these are the new ones. This is the oldest one. You might say the oldest one is at the base. No, the new one is arising. So these are the new ones. New are arising from the base of the old one. So new, this is the older, so new one is arising from the base of the previous one. So that is why we call it basipetal arrangement. Are we clear till here? Are we clear? Please tell me. Yes or no? Now guys, moving on to the flower. So flower, in flower guys, the leaves have modified into floral appendages basically. So what is a flower? Flower is a modified shoot. It is a reproductive shoot and it is a modified shoot, right? Where basically the leaves change into floral appendages and internodes will condense, right? Internodes will condense on thalamus. They'll come very near to each other. Internodes condense on thalamus. So the four whorls we know: sepals, petals, stamens, pistil, and these are the collective terms. All the sepals are collectively known as calyx. These are called corolla. These are called androecium, and pistil or carpel collectively known as androecium. Is that clear? Now guys, now flower can also have a symmetry, and it does have a symmetry. Just like in case of animals, we divide them into two equal halves. Flowers will also be divided into two equal halves. Is that clear? If they can be divided into two equal halves, then symmetric. If they can't, they'll be known as asymmetric. Now the asymmetric ones, the example of asymmetric is kanna. No matter which plane you use, it cannot be divided into equal halves. Then we have actinomorphic. So actinomorphic basically means radial. It means radial. So like in case of animals, radial symmetry is the one where organism can be divided into two equal halves along any plane. Same is the case here. Along any plane, you can divide them into two equal halves. And then bilateral, also known as zygomorphic, along only one plane, they will be divided into two equal halves. Is that clear? So we symbolize this as any plane. Plus means either you cut it like this or you cut it like this, in any plane. And this one, zygomorphic, only in one plane, there will be two equal halves. Is that clear? Now, important part is to learn their examples. See here, how do I learn? So I have learned it as, so see, you can learn it as Dhatura must act chill. This is a complete mnemonic. Dhatura must act and chill. So chill is chili, must is mustard, and Dhatura. And then zygomorphic, that is Zyghicacia. So how do I learn? Ziggy is for zygomorphic. Zyghicacia ne gulmohar pee ke been bajai. So this is how I learn it. So these are the examples. Please tell me these points are clear. Yes guys, please tell me these points are clear. Right? Now moving on to the next part, which is different types of whorls. How many whorls are present? So see, if sepals, petals, and androecium, they are present in the multiple of 3, then we'll call them trimerous. 3 and multiples. 3, 6, 9, and so on. If they are present in the multiples of 4, then 8, 12, and so on, tetramerous. And then if they are in the multiples of 5, then they are pentamerous. Not very important here, but just be able to understand the word meaning. Now guys, we have types of flowers on the basis of the position of ovary. Now on the basis of position of ovary, we have 3 types of flowers. One is hypogynous, then perigynous and epigynous. Now this is very, very important. There is already a PYQ that you can see here. And the diagram based PYQ also. So PYQ can come up in 2 ways, diagram based as well as example based, and third, definition based. Now see, so in hypogynous flowers, hypo means below, gyne means ovary. So all the other structures will arise below the ovary. So ovary will hold the topmost position. Ovary is at the topmost position. So such ovary is known as superior ovary, guys. So remember, hypogynous will always have superior ovary. Then talking about perigynous, peri means around, gyne means ovary. So everything is going to arise at the same level as that of ovary, and it depends upon the type of thalamus actually. So if this is the shape of the thalamus, ovary will have the topmost position, right? Rest everything arises below here, below the ovary. Now in this case, peri, all are going to arise at the same level because such ovary is called half inferior. When it is sitting in parallel position to rest of the structures, we call it half inferior. And then we have epigynous, guys. Epigynous, epi means above the ovary. So all the structures will be present above the ovary because this is the shape of the thalamus. So the ovary is going to go here. So if everything, in this case, all the other structures will arise above the ovary like this. Point clear? So in epigynous flowers, ovary is holding the lowest position, therefore ovary is inferior in nature. Please tell me these points are clear to all. And now the most important part of this topic, which is the learning the examples. Yes, guys. Okay. Let's see here. So hypogynous. So we have he must bring superior China rose. So this is how we learn. He for hypogynous, superior is for superior ovary. Example is mustard, brinjal and China rose. So for perigynous, guys, so people pluck peach and inferior peach, people pluck peach and inferior rose. Half inferior, people pluck half inferior peach and rose. So people means perigynous, plum, peach and rose. These are the examples, and half inferior is the type of ovary. And then we have epigynous. So epic sunny cooks, inferior guava. So we know epic is epigynous, sunny is sunflower, the ray florets. Then cooks is cucumber, that is the second example, and third is guava. So this is how we learn the examples of types of flowers on the basis of position of ovary, guys. Are we clear? And then we have estivation. Now what do we understand by estivation? Estivation is the arrangement of different structures in the same wall. Is that clear? So this is arrangement of structures within the same wall. That means how is the arrangement of petal, one petal with respect to the other, one sepal with respect to the other sepal. So you don't crisscross the whorls. So sepals to sepals, petals to petals. So that is how we find out the estivation. So we have 4 types of estivation. One is valvate. So guys, in valvate estivation, see, there is no overlapping. There is no overlapping. So it's like margin to margin contact. Margin to margin contact. No overlapping. Is that clear? They might be fused. If they are fused, this will show fusion. If they are not, then we will not put this structure. They can be fused, but there is no overlapping. This is valvate. Now second is twisted. So in twisted, guys, you see one of the margin is out, another margin is inner. One is out, another is inner. One is out, other margin is on the inner side. One is out, other is on the inner side. This is called twisted. So, right, this is twisted. Then in imbricate, there is no fixed pattern. It can go in any direction. And we'll talk about vexillary. First, let's see the examples. So, valvate, CV, calotropis. How do I learn? Culval. Can you see? It is culval. Then, for twisted, I learn it like cotton, China rose, ladyfinger. And then waxillary. This is a characteristic feature of Fabaceae. Fabaceae, peas and beans. Clear? And then imbricate. This is found in Gulmohar and Acacia. This is GIC. So if any one of you is ever interested in dentistry and ever, or you have a sibling in dentistry, then you must know what is GIC. If not, then never mind. Maybe you will find it someday or not. Never mind. Just remember GIC, guys. So GIC is Gulmohar, imbricate and Acacia. Now, talking about waxillary, waxillary is also known as Papilionaceous. Papilionaceous because it is found in the subfamily Papilionaceae of Fabaceae. Papilionaceae. Is that clear? Now let's see how does this look like. See, in Papilionaceous of waxillary condition, guys, there has to be 5 petals only. This is fixed. This is fixed. Now there is this largest one. This largest one, largest petal is known as, standard. How many? Single. Then there is smaller one which is called lateral. These are the laterals. One margin in, other is out. Then we have the smallest ones known as keel. These are 2, and do you see this shows their fusion? They are fused. They are also known as wings. Is that clear? Now, when we are talking about flowers, the brackets are very important. Brackets have a meaning. Parenthesis. The brackets mean you are showing fusion. So there is 1, there are 2, and then there are 2. So last 2, that is the keels, they are fused. They are fused. Please tell me these points are clear. Yes, guys. And you also have to note down that keel are present on the anterior side. This is another question, guys. Keel are on the anterior side. Please tell me. Yes, guys, please let me know. Quick, quick, quick. Are we clear till here? So there is this question that standard is present towards which side? So there is a question. If you are solving PYQs, you will be able to see. Otherwise, we will be practicing them in the class also. And I have discussed this question in my special classes before the NEET also, and now also I'll be discussing that. So remember, it is SP and this is AK. So P and S come together and A and K are nearer. No logic at all. Just remember, this is towards the anterior side, that is towards the posterior side. Is that clear? Please tell me these points are clear to all. Now guys, we are going to discuss some conditions here regarding petals and sepals. See, let's talk about these conditions and their examples. So, talking about sepals. If the sepals are fused, we use the term gamosepalous. We use the term gamosepalous. Gamo means fusion. If all the sepals are free, they are not attached to each other, we call them polysepalous. Similarly, if the petals, together known as corolla, they are fused, gamopetalous. If they are free, we'll call it polypetalous. Now, talking about stamens, collectively called androecium. See, if they are sterile, we call it steminode. This was 2024 question. If they're attached to the petals, we call them epipetalous. We call this condition as epipetalous, 2024 question. Then, if they are attached to the perianth, we call it epiphyllous. Now, before you ask what is epiperianth, now there are times when calyx and corolla are indistinct. When calyx, sepals and petals, calyx and corolla cannot be distinguished, they are indistinct, then we don't use the term calyx and corolla for it. Then we call it perianth. and the individual unit of perianth is called tepal. Now, in case stamens are attached to the perianth, then the term is called epiphyllous. This is in lily. Epipetalous, is that clear? Is when it is attached to the petal. Clear? Then talking about gynoecium. So see, gynoecium, if they are free, we'll use the term epo here. Unlike here, for free we use the term poly, and for fusion we use the term gemo. Here we are using the term epo for free, right? And syn represents fusion. So epocarpus, syncarpus. Are we clear? Is that clear? Yes or no? Now, just like the free and the fusion case of gynoecium, androecium can also be free or fused. See, now see here, if the androecium are free, we use the term polyandrous. If they are fused, we use the term synandrous. Okay? Let's say there are 10 stamens, they are all free, we call it polyandrous. They are together going to form bundles, they are going to bundle up then. So in cylindrous, if they are bundling up to form one cylinder, we use the term mono. So one cylinder they form, one bundle they form, we call it monoadelphous. Is that clear? If they bundle up to form two bundles, two cylinders, we call it diadelphous. And if they form more than two bundles, we are going to call it polyadelphous. Is that clear? Now see. So as I said, bracket means fusion. So if they are 10, we will simply write A10. If they are fused, I am going to write A10 fused. All the 10 fused to form one bundle. This symbol represents synandrous condition and monoadelphous condition. Then if I write A5 plus 5, it means it is of course synandrous, but then how many bundles? 2 bundles, and each bundle has 5. Or it can be A9 plus 1. So there is no bracket around 1, of course. This is wrong. So how do we write this? A9 plus 1. Is that clear? And then polyadelphous. This is just haphazard, just trying to show how it can be present more than 3 bundles. Is that clear? Then talking about the fusion in case of pistil. So if the pistil is 1, and it will call it monocarpellary, right? So 1 will obviously be free. So we use the term epocarpus. But if there are 2 pistils, they can be free as well as fused. Bicarpellary for saying that there are 2 pistils. Now these 2, if they are fused, we'll call it syncarpus. If it is free, we'll call it epocarpus. So there can be bicarpellary and epocarpus condition, and bicarpellary and syncarpus condition as well. Please tell me these points are clear to all. Yes guys? Please tell me these conditions are clear to everyone. Now another very important topic, and that is placentation. We have placentation. Now guys, what is placentation? Let's understand. So placentation is the arrangement of placenta. What is placenta? So we know that this is ovary, right? And in ovary we have some seeds. So seeds or ovules are not directly attached to the ovarian wall. So there has to be a cushion-like structure which is called placenta. So how many placenta and how these placenta are arranged within the ovary or ovarian cavity, this arrangement is called placentation. It is the arrangement of placenta. And wherever there is placenta, there is an ovule. So you can say arrangement of placenta or ovule within the ovary. Is that clear? Now, there are 5 different cases that we have to discuss: marginal, axile, parietal, free central, and basal. Now see, first of all, the examples are very important, and then their characteristics. See, in marginal, guys, what happens? So it is present in case of P. So we see that there are two. So there is a ventral suture. There are two margins, ventral margins, and where the two margins combine, on each there is present a ridge. See, on the ventral sutures, marginal ovules are present, and it is simple unilocular ovary. So ovules are present on the ridge. They form a ridge. Is that clear? This is where the ovules must be present. So this is the placenta, and on the placenta are present the seeds or the ovules, like this. Are we clear? This is marginal. Seen in case of pea, or you can say beans, this is marginal, right? Then talking about exile. So in case of exile, guys, there is a central column, multilocular ovary, and multilocular ovary is divided by septa. So septa is present, true septa is present here. So that is in case of tomato, lemons, and China rose. Parietal, this is what parietal looks like, all around the ovarian wall. So ovule at the inner wall of ovary, and there is presence of false septum. Just these are the key words, guys. So false septum word is used for parietal, and this is in case of argemone and mustard. And then free central. What happens in free central? So there is a placenta in the center, and then there are no septa present. So here septa are absent. See there, with respect to septa, true septa in exile, septa absent in free central, and parietal has the false septa. So what happens if this is a placenta, guys? If this is a placenta, around the placenta, this is how the ovules are present. This is primrose and dianthus, an example of free central, right? And then in basal, we have marigold and dahlia. So sunflower, not this. This is sunflower and marigold. We have basal. So it is at the base. So how is it present? Like this. So this is the ovary. At the base, we have one placenta, and on this we have the ovule. So this is basal. So learn the keywords also. Marginal, ventral suture, unilocular ovary, ridge. These words are all associated to marginal. Then true septa. So multilocular ovary, right? So this is all exile. Then parietal, we have this word false septa. And then free, we have no septa. And in basal, it is just one ovule at the base. Please tell me these points are clear. Yes, guys, please tell me these points are clear. Quick, quick, quick. Are we clear? Are we clear? All right, guys. Moving on now to the next, we have fruit. Now, flower will become a fruit after fertilization. So, guys, we have three different types of fruits. First is a true fruit, then false fruit, and then we have the third type called the parthenocarpic fruit. Now, what is a true fruit? True fruit will develop from ovary. If that fruit develops from any part other than ovary, it is called false fruit. And in case of parthenocarpy, there is no fertilization, so there will be no seeds. So these lead to the formation of seedless fruits. Let's talk more about true fruit, which develops from ovary, like guava, mango. False fruits. Now, part other than ovary will develop, and it is usually thalamus. For example, apple, cashew, strawberry, these are all false fruits. And as I said, these are seedless, so parthenocarpic. And these days, let me know in the comment section or in the chat, which is that hormone which helps in inducing parthenocarpy. For example, tomatoes. Quickly let me know. Let's see how much you are learnt. So we are revising, but are you revising, guys? Quickly let me know which is that hormone which helps in inducing parthenocarpy in the fruits. Quickly. Auxin. Very good. Very good, Kavya. Nice. So that is auxins, right? Now, talking about the seed. See, flower will become fruit and ovule will become the seed. So it develops from the, or can we say seed is a fertilized ovule? Like fruit is a fertilized flower, sorry, ovary, and this is a fertilized ovule. Right? Now, what does seed consist of? There are two parts, two things. One is the seed coat. Outer one is called testa, other one is called tegmen. Now, which part will actually develop into which part of seed? We will discuss this in the coming up session of part 1 botany, that is sexual reproduction in flowering plants. So that is the chapter where we actually get an insight how these structures develop into or modify into the other structures, right? So wait for that. And second is embryo. So embryo has three further parts: radicle, which will give rise to root, then embryonal axis, and then cotyledons, which can be one or two. If there are two, that seed will be dicot. If there is one, that seed will be a monocot. And so guys, before we move on to the next chapter now, let me remind you, as I've told you in between also, that apart from Re-NEET preparation, 2027 aspirants should stay very focused, and for that the batches have already been, the batches will be starting very soon, like after Re-NEET, but those already in class 12, the batches have started. So guys, don't forget to join in case you are late. And these dates have been shifted. Because of Re-NEET exam. But quickly enroll so that we can start with the preparation. My classes have already begun. Is that clear? Now, moving on guys, moving on to the next chapter, which is cell cycle and division. So guys, we are going to discuss this chapter. See here, I'm going to discuss the entire chapter in the form of the flow chart. First of all, see here, we know every cell cycle, be it mitosis or meiosis, cell cycle is divided into two phases, which are number one, interphase, the preparatory phase or the resting phase, and the second one is the mitotic phase, right? Now, we know that this interphase, so from here to here, this is all interphase, yes? Now, in this interphase, actually the cell is going to prepare itself for the preparation. Now, actually, wait, yeah, okay, now see here. So in reality, the actual division takes just very less time. Okay, so 95% of the time is taken by this interphase. This is interphase. And interphase was earlier known as resting phase because nobody could see them working so hard. So nothing major happened during, like nothing visually happened, nothing visual was happening during the interphase. So we called it resting phase, but this name is actually a misnomer. So interphase, and we also call it a preparatory phase because it is during this phase the cell is actually preparing itself for the real division, and it consumes 95% of the total time. For example, if it is a human cell which divides once in every 24 hours, so this is going to take 23 hours out of 24 hours in case of human cell. And in case of yeast, if yeast is dividing every 90 minutes, like 1.5 hour, then you just calculate how much 95% would be in case of yeast cell. Is that clear? Now see, interphase is further divided into three phases. We have G1, we have S, and G2 phase, right? G1 is the growth one or gap one phase, and this is the real division phase. This is the real division phase, which is called the M phase. Yes, this is called the M phase or the real dividing phase. Now, in division, basically, again, division will occur in two parts. First, it is the nuclear division, followed by division of cytoplasm, right? So, we have karyokinesis, another term for nuclear division, and we have cytokinesis, term used for cytoplasm, division of cytoplasm. Are we clear till here? Now, the karyokinesis is further divided into PMAT, prophase, metaphase, anaphase, and telophase. Yes, and then we have cytokinesis, right? Now, this is how the life goes on. The cell keeps on dividing. So after every division, it will go in rest and prepare itself for the next division and again divide and so on. This is the ruckus of life. Okay. Now, there are times when the cells, they plan to exit this Mohamaya, right? They plan to get out of this division. So, they exit through G1 and get into another silent phase, G0 phase. And this is where they acquire specialization. So, this is the phase of specialization. We also call it quiescent phase. This is the phase where they actually acquire specialization, they become differentiated, they become mature cells, and they become functionally active cells. They are able to do something other than just division. So all the cells in your body, be it skin cells, your heart cells, your eye cells, they are basically all in nerve cells. They are all in the G0 phase, where they have become specialists. So you have to get out of a ruckus to become some specialist, right? Now, are these points clear? Now see here, once again. So what are the events which will take place during G1 and S and G2? See here. So G1 goes for 11 to 12 hours, S for 6 to 7, G2 for even lesser. Now, see, in G1 phase, what are the things happening? In G1 phase, there is cell is growing, there is multiplication of the cytoplasm and all those things. And then in S phase, the characteristic of S phase or the synthetic phase is DNA multiplication, histone formation, centriole multiplication. Is it clear? What happens during the G2 phase? So protein synthesis. Not all proteins, but NCERT writes that protein synthesis occurs in G2. So basically tubulin protein. Organelles duplicate. More RNA synthesis takes place. I've already written here. Okay. So this is protein synthesis occurring in G2. So you have already seen PYQs based on the events occurring in G1, S and G2. Please make sure that you are ready for similar questions for Re-NEET and for 2027 as well. Is that clear? And see, in G0 phase, cell remains metabolically active, but it undergoes differentiation. Are these points clear to all? Yes? Okay. Now, what happens during the prophase? Your nuclear membrane, nucleolus, GB, lysosomes, all these structures disappear. Centrioles are going to move away towards the poles. Chromatin fibers will condense to form chromosomes and mitotic apparatus will develop. So what is mitotic apparatus? It is the spindle fibers and the two asters. Make sure that you are revising these things. I have compiled the entire NCERT in this one cyclic flow chart. Is that clear? Then talking about metaphase. This is the best stage to study chromosomes. Equatorial plate is formed. Now equator, just like equatorial plate is also a imaginary plane. Okay, so that one plane where all the chromosomes are going to align themselves is called the equatorial plane. And now guys, this is the best stage to study chromosomes. What if somebody says, which is the best stage to study the structure of nucleus? That is interphase. Is that clear? Best stage to study the structure. Best stage to study the structure of nucleus. That is interphase. Is that clear? But best stage to study chromosomes would be metaphase. Now here spindle fibers are attached to the centromere through the kinetochores, and then once they are on the equator, spindle fibers attached, now there will be pulling of these centromeres. The spindle fibers are going to contract. So this will lead to splitting of the centromere and separation of the sister chromatids, and because the spindle fibers are contracting. In telophase, guys, this is just the reverse of prophase. So here, all the structures which disappeared, they will reappear here. Now, chromatin was condensing to form chromosomes here, but here they will decondense to form chromatin fibers. Is that clear? And cells will have two daughter cells. Please tell me these points are clear to all. Right? Now, last not the least, now when the nucleus has already divided into two daughter nuclei, it's time for the cytoplasm. Now see, in animal cells, the division occurs through furrow formation or cleavage. So this is called centripetal fusion, centripetal cytokinesis. While in case of plant cell, guys, there is a cell plate formation. It is basically Golgi body vesicles coming together in the center and then expanding outwards. That vesicle collection is known as phragmoplast. So since the plate is going to move outwards, this is called centrifugal cytokinesis. Are we clear till here? Please tell me. Are we clear? Yes, guys? Quickly let me know, are we clear till here? All right. Now, moving on, guys, moving on, there is another aspect here to be noticed. Do you see I have written some cases? See, if in the beginning, guys, the parent cell had chromosome number 2 chromosomes, and let's say the DNA content of 2 chromosomes is 2D. What is the chromosome number in S phase? After DNA duplication, the chromosome number will not change. It is only the DNA content that will duplicate. Why? See here. If here there were 2 chromosomes like this, not chromosomes actually, they were in the fibrous state, chromatin fibers, but let's say there are 2 like this. The content of each is DD, so hence 2D and the 2 chromosomes. Here also, after DNA duplication, the chromosome number will remain same, but the DNA content will double. Why the chromosome number remains same? Because now here the centromere is the same, and it is around the same centromere that sister chromatids have developed. Please see here actually the sister chromatids develop. So sister chromatids are formed during the S phase and they will separate during the Ana phase. Are we clear? Please tell me. Right? So guys, the trick is the number of chromosomes is equal to the number of centromere as long as the centromere is the same. Even though there might be more sister chromatids, still the number of chromosome will not change here. DNA content will go on increasing, but the chromosome will remain the same as long as centromere is the same. Please tell me these points are clear to you all. Is it clear? Now, what happens in the G2 phase? Nothing significant about chromosome number and DNA, so that remains the same. In prophase also, guys, chromosome number remains same, 2 and 4D. In metaphase also, chromosome number remains the same, 2, and DNA also remains 4D. Now, anaphase is the shortest phase, and see what happens. Because there is splitting of chromosome, now the chromosome number will double up here. The DNA content will remain the same. Now, because there is splitting of centromere, now instead of 2, there will be 4 centromeres, right? So number of chromosome is equivalent to number of centromeres. So here we see the number of chromosomes doubling up. I hope this point is clear to all. And now the same remains in telophase, and then after cytokinesis, the chromosome number and DNA content in each daughter cell, guys, is restored, and this is exactly same. See here, it is exactly same. This and this, it is exactly same. So that is why, guys, this is a case of mitosis where the number remains same, equational division. Please tell me these points are clear to all. So in this flowchart, guys, we have talked about the chromosome number and how it changes, and we've also discussed about the major events which are occurring during each phase. Now, time for meiosis. See here, we'll take one thing at a time. Okay, we'll take one thing at a time. Now, see here. Okay, so from here, guys, from here to here, I have tried to represent the interphase. Now, interphase is the same as in case of mitosis. The same type of events are going to take place in here, G1, S, and G2. Is it clear? But now the M phase is divided into two meiosis. One, we have meiosis. So in first phase, we have meiosis 1 till here. We have meiosis 1. This is meiosis 1. And then we have meiosis 2. Are we clear? Are we clear? Now see. In meiosis 1, see here, in meiosis 1, the prophase is the longest phase. And it is further divided into leptotene, zygotene, pectine, diplotene, and dikinesis. Right? So these 5 substages form prophase 1. Is that clear? Then we have metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. It may occur immediately or it may occur together, the cytokinesis may occur at the final stage only. But let's stay simple, take it normal case and continue with cytokinesis 1 immediately after meiosis 1. Right? Then there will be a short break between the two meiosis 1 and 2, which is called interkinesis, and nothing significant happens. Then this is prophase 2, metaphase 2, anaphase 2, telophase 2, and then second cytokinesis. Are the stages clear? Now let's talk about the events that are going to take place during each phase. Told you already, interphase is just the same like we have studied in the previous chart. Talking about prophase 1, see, in prophase 1, just like in mitosis, nuclear membrane will disappear, organelles will disappear, those things are happening, disappear. Centrioles move polewards, chromatin becomes chromosomes, mitotic spindle appears. This is going to occur during the entire prophase 1. Right? Now, why have we divided prophase 1 into 5 stages? Because some characteristic things were happening here. For example, see, in leptotene is also known as bouquet stage or spirene stage. So here basically all the chromosomes or the chromatin fibers, they are in the bouquet stage. So they are converging on the lower side and they open up like a bouquet. So it is because of the arrangement. And here, since they have started condensing, though they appear like beads on the string stage. So basically nucleosomes will start becoming visible, right? Beads on the string stage. Then in zygotene stage, guys, synapses is going to occur. So synapses means the two homologous chromosomes are going to lie side by side. That is synapses, right? So for the two chromosomes to lie side by side, there has to be a formation of a nucleoprotein or proteins, so not nucleoprotein. There has to be a formation of fibrous structure, and this fibrous structure is made up of proteins, right? And that fibrous structure is called synaptonemal complex. Synapse means lying side by side, nema is thread. So thread-like structures are going to keep the two homologous chromosomes together. So when the two homologous chromosomes are lying side by side, guys, like this, so this is one, and then there is another, and then there is a synaptonemal complex in between. This is called synapses, right? So this stage is called bivalent stage, and since there are 4 chromatids together, we call it a tetrad stage. Two homologous together, so bivalent. 4 chromatids, so tetrad stage. Are we clear? Right? Then what happens during Pachytene? So in Pachytene, crossing over will take place. So Pachytene is characterized by crossing over. So when the crossing over occurs, there is recombination nodule formed for the and enzyme used is recombinase. And remember, the crossing over will always occur between non-sister chromatids of the homologous chromosomes. Very important to remember. It will occur between the non-sister chromatids of homologous chromosomes. Are we clear? Which are the non-homologous chromosomes? So it's this one and this one. These are the non-sister chromatids but of homologous chromosomes. Are we clear? Are we clear? Right? Then coming on to diplotin. Diplotin is opposite of zygotine. So here the chromosomes were brought together by synaptonemal complex and this is known as synapses. So in diplotin, there will be dissolution of synaptonemal complex. When the complex will dissolve, the two homologous chromosomes will try to run away from each other as if they never wanted to come together. So this is called desynapsis. And they will only remain attached at the point of crossing over, and that only attachment is known as chiasmata. Chiasmata is an X-shaped structure which is the site of crossover where they are still attached. Please tell me these points are clear. And last not the least, finally the chiasmata, this point of attachment, will also detach. So gradually this chiasmata is going to move towards the tip and gradually it is going to separate. So terminalization of chiasmata occurs during dikinesis. So we have already seen many questions around this topic in the format of match the following. Yes? Now coming on to metaphase. Here in mitosis there was one equatorial plate formed. In metaphase 1 there are two equatorial plates formed. And what will happen? Each chromosome will be present on either of the two plates. Out of the two, chromosome number 1. See, we are diploid. We have 2 copies of each. So one copy will be on the upper plate. Other copy will be on the lower plate. Similarly for each. So one out of all the one out of the 2 pairs of chromosomes. Sorry, each pair of chromosomes. One will be on the upper equatorial plate. Other will be on the lower plate. Are we clear? And spindles are attached to the kinetochore. Please tell me these points are clear. And then anaphase, there is no splitting. In anaphase of mitosis, there was splitting of centromeres, separation of sister chromatids, but not in this case. In this case, the whole chromosome on one equatorial plate will go towards one pole and the other chromosome will move towards the other pole. So no splitting of centromere. There is instead a separation of homologous chromosomes. Are we sorted here? Yes. And then guys, again, telophase 1 is going to be reverse of prophase 1. So all these structures which disappeared, they are going to reappear here. Chromosomes will again decondense for a while. Is that clear? So this is what happens during meiosis 1. Now it will be followed by cytokinesis and give rise to 2 daughter cells here itself. And then each daughter cell will enter into the meiosis 2. And now meiosis 2 is exactly like mitosis. So treat as if each daughter cell is going to undergo mitosis. Right? Now coming on to the very important aspect for the numerical purpose. That is the number of chromosomes and the DNA content. Let's assume the same part. Let's assume the same thing here as well, that there were 2 chromosomes in the parents and the DNA content was 2D. So after S, we know the chromosome number does not change, but the DNA doubles. Now same continues in the G2 phase. Now the same thing will continue in the entire prophase. In the entire prophase, this is going to remain same. Chromosome number will remain same and DNA content will also be same as that of S phase. Right? Here also it will be 2 and 4D. Here also, since the splitting did not occur, so chromosome number also remains the same and DNA content also remains the same as after S phase. So you have to note down the words very carefully while solving the questions. after S phase, I said. This is what the language of question is. You have to compare the two stages. So they've often written the amount of DNA in a cell after it has undergone S phase. Are you getting that? Right? Now see here, in telophase, again same, 2 and 4D. Now after meiosis 1, see, each daughter cell will have half the chromosome number, half the DNA content. Now because the chromosome number is reduced to half, guys, that is why meiosis 1 is called the reductional division. That is why it is called the reductional division. Please tell me these points are clear to everyone. Are we guys listening, or are we talking about biryani? Yes, please. Who's so hungry for biryani right now? How can you think of biryani? Really? Now eat biryani the day you get your paper done right, on 21st of June. Till then, survive and study. Now see, now each daughter cell enters the meiosis 1, and see, you will treat it as mitosis, right? So here in the beginning we have chromosome 1 and DNA is 2D. In metaphase again it remains the same. Now in anaphase 2C, in anaphase 1 there is no splitting of centromere. In anaphase 2, splitting of centromere will take place. So when we know when the centromere splits, the chromosome number will increase. So here it was chromosome 1, here the chromosome number will become 2. The same happened in mitosis. Then chromosome 2 and 2D, and then it will further divide. So each chromosome cell after the meiosis 2 will have 1D and 2D. Yes. Now compare it with the parent cell. Compare it with the parent cell. Parent cell had 2 chromosomes and 2D DNA. This will have 1 and D. Question is, usually after, now compare the daughter cell which has undergone meiosis 2 with the cell which has already undergone S phase. Now if I compare it with this, what is the change in the amount of DNA here? Can you tell me? If I compare the daughter cell after meiosis 2 with the cell, parent cell after S phase, if I compare these two, what is the change in the DNA content, guys? Quickly let me know. If you can answer this question, you can easily crack NEET. But if you are thinking about biryani right now and just chatting, then I have doubts. Yes, guys. Quickly let me know. What is the change in the ratio? Quickly. Do we see here it is 4D, here it is D. So the amount of DNA has reduced by 1/4. Yes, it has reduced by 1/4. So guys, with this we complete cell cycle. Now talking about the chapter of cell structure, I am going to record a 15-minute video for cell structure, and we are going to discuss that cell structure in that session, because you've seen something has happened to the PDF and my flowcharts are gone. So I'm going to redo that and make a separate video of cell structure. Don't forget to watch that and complete that. And also about plant families, 6 families that we have to do. I've already made the quickest video possible in which we can learn the families in the best and the easiest way possible. Is that clear? So how to learn families, that video is already done. Please watch that, and I am going to discuss questions around these topics now from maybe tomorrow onwards in special classes as well as here. So I am going to bring, so my class 11th is done, guys, both zoology and botany. So with this we complete our class 11th. So after this, guys, now we are going to start with class 12. So be ready for that. But before moving on to class 12, let's make sure that our question practice for class 11 is thoroughly done. And also, once again, coming Sunday and Wednesday, you will have your All India mock test. Make sure that you enroll in it and you take the test. If you are still not taking these all India mock tests seriously, then guys, this is a waste of great opportunity which sadly has come in front of us, but let's make the best use of it. So thank you so much. I'll see you in the next session and we will be starting with class 12 biology. But before that, I'll see you in between and we'll be discussing some very important things. Is that clear? You'll get notified, so no, don't forget to hit the bell icon, join the Telegram channel, and also guys, you will get these notes, you will get all these notes on my Telegram channel, which is Tina Ma'am Biology. Is that clear? I'll upload these notes right away and I'll see you in the next class. Thank you so much. Don't forget for AIMT and I'll see you in the classes. Bye.